go to full screen. I'm having the same, yes, okay. This is on yours, not on mine. Huh. Got two screens open.
Oh, that's the presentation.
Okay. All right. I want to pick up here. We left off last class. Did people get a chance to look at the video? I know it got up late or not yet. Some? Yes? No? Not so. Okay. All right. So we left off in here, remember, for around Tuesday, right? With about factors. Okay. So let's go back a slide. Okay. All right. So we're just talking here quickly. We'll just do this very quickly. They rehash, right? So term ones up front. Normal flora, normal microbiota, right? So the correct term is normal microbiota, which again is just, you'll hear me say normal flora all the time. You know what I gotta put on in a minute? It's that microphone. But anyway, okay. Or is it over here?
Oh, I think you have it.
Oh, I have it. That's right. Okay. Well, people, I'm not used to having to do this, right? This way. You just come in here, we blab. Oh, this stuff. Good? Normal flora, normal microbiota. I'm going to say normal flora, technically it's normal microbiota. They're the bugs that live on your skin, right? And other places, not just your skin. How about your pharynx, your throat? You have any bugs in there? It's loaded, right? And I made this point in the other video last week is our mouths, cats' mouths have about 10 to the 9 organisms per milliliter of saliva. Right? Dogs are a little further down the list, but our mouths are loaded. How about your lungs? Your lung fields beneath the epiglottis, right? When you get into your lower lip, lots of bugs there. Not really, not so much, right? We used to teach it sterile. It's not really completely sterile. I mean, sterility, of course, means what? There's no infectious agents, no spores, anything like that. But that's not totally true. But generally, we don't have a lot of bugs in our lungs. If you get organisms in your lower respiratory tract, what do you get? Pneumonias, exactly, right? Okay, so, and that's really, but our mouths and our upper respiratory tract is loaded. How about your colon? You have a lot of bugs there?
Yeah.
You have 10 to the 12 bacteria per gram of your lovely feces, okay? All right? Gazillions of them, all right? Now, certainly, hopefully, in your joint spaces, like your knee, right? Things like that, elbow, there's no bugs there. You don't want a lot of bacteria in your blood, all right? It happens. Did you brush your teeth today? Of course you did, right? And you flossed as well. Your last name is?
My last name? Faminu.
Faminu?
Mm-hmm.
Of course she did, as Faminu did, right? But as soon as she was brushing your teeth or she flossed, what happened? Bacteria got into your blood. But your immune system cleared them quickly, right? So, again, your blood's usually pretty sterile, all right? Believe it or not, when you defecate many times, right? And I know none of you have a problem with constipation, right? But when you really, some people go in there, they bring a book, they read for 30 minutes, right? You can hear them across the hallway, right? Those people, by the time you're done, they've actually, bacteria have seeped into their bloodstream too, okay? So what happens? It's transient. Our immune system clears it pretty quickly. So we got lots of bugs all over us. Okay. And of course, just the point I made in class was that infectious disease deaths worldwide, 24.7% of all deaths, number 2 behind cardiovascular disease. Okay. And then among the infectious diseases, about 10% are respiratory and gastrointestinal. So respiratory is part of what we're doing here. These masks aren't going to do much to stop incoming virus, but they will help outbound respiratory droplets, okay? Which is a 1 meter kill zone. And the extra few feet is to help prevent transmission. Gastrointestinal is harder. They're both hard to control. Respiratory is hard to control, and so is GI. You know, think about it, right? But many people do not wash their hands. They're going to do their thing in the bathroom, all right? And then they leave, look right, look left. Half these people never wash their hands. They're going to touch those fomites, those door handles. They want to touch your face, right? Okay? They want to borrow your cell phone, okay? And what do? They take all these organisms, right? And they put them on a fomite, an inanimate object. You know, you touch it, and then just innocuously, you rub your nose, you itch your face, right? Or your eye, whatever, and you all of a sudden have a gastrointestinal bug. Hepatitis A, right? All sorts of bacteria. That's a virus, but I mean, all sorts of stuff gets transmitted. So again, respiratory and GI, about 10% of all deaths. They're really hard to stop, okay, overall. Okay, and of course, with respiratory, we try to do what? Well, the mask can help, social distancing, things like that. Okay. And then certainly, again, right, just some of these organisms are absolutely pathogens, right? Word path is disease. So we'll talk about primary pathogens in this course, which are diseases like tuberculosis, okay? When you inhale the tuberculous bacillus, means just a rod, you're going to get TB. But the good news, and we'll get to this in detail about week 9 or 10, about 10. With TB, what happens though is 90% of us, 95, will clear the infection, but you're going to get infected. It's a primary pathogen. But if you live with someone with TB and you get infected every single day, right? Now, Miss King, if she has active TB and I'm around her every day, and sooner or later, what's going to happen to me? Every day I get infected. What's going to happen? Sooner or later, what? Your luck runs out, right? It's like Russian roulette. Sooner or later, you're going to lose people, right? Okay? Same sort of thing. That's how TB is. It's a primary pathogen. Okay. All right? So other bugs are opportunistic pathogens. These are bugs, again, we use the skin as an example. You got lots of organisms on your skin. And many times there's a bug that about a third of you have in your anterior nares, that's the front part of your nostrils here, all right, called Staphylococcus aureus. Okay, and staph, again, 1/3, 30 to 40% of you have it right now, right, in your anterior nares. You're colonized, okay, and it doesn't mean you have an infection or you have a disease, but you're colonized, okay? How about me? What's my rate of colonization? Probably 100%. I'm around sick people with nasty infections every day, right? So I get colonized all the time. I don't drill a lot. I try not to do that, okay? But it happens. But if we took a Q-tip and swabbed any of you, you'd be positive for Staph aureus, all right? So it's hanging there, you're colonized, but if you get a cut in your skin, or, right, you decide to go drilling, all right, and you end up damaging your tissue, then the staph becomes a opportunistic pathogen, okay? It takes over. So we have lots of bugs this way. So part of our normal microbiota, they're your friends, okay, but also they are opportunistic pathogens. Given the chance, they'll nuke you, all right? And that's just the way it works. All right. But again, what do they do on our surface of our skin? Well, again, for the skin, or our upper respiratory tract, or your GI tract, they take up space, they use up nutrients, and they make it tougher for some of these other pathogens, disease-causing agents, to get a grip, right, on your tissues, your throat, your skin. Okay? The analogy I like to use is MARTA, right? Who's been through Lindbergh Station 5 o'clock on a weeknight? Nobody rides MARTA? Never been to Lindbergh Station? Right? The trains are packed. Maybe not now, but usually, a normal pre-COVID day, right, they're packed. You have to cram people in to get in. That's like your normal flora, right? The trains are packed, the pathogens are trying to get in. So they have a great effect for us. Okay, so in this class we're wrapping up here. We went through some of the syllabus and stuff like that. And we'll do a few questions from that today. But we sort of picked up 3 big technological advances that have changed a lot in our lifetimes. Sanitation, vaccination, and antimicrobials. So for sanitation, of course, we talked a little bit about, right? Nobody here dumps their feces right outside the window anymore, okay? Now, you go to some undeveloped countries, it's still pretty common, right? People don't do a lot of great, they don't have necessarily the sewage systems we have. But certainly here, we clean things up quite a bit, and that helps decrease a lot of the fecal-oral type transmission. But all you need is what? A good hurricane, a good earthquake, right? What happens? Sewage and water can get mixed up pretty easily, lines break. If you live in downtown Atlanta, every year what happens? Some of the older water mains break, right? And they ask you to boil your water for a couple days, right? Because they try to figure things out. Okay. All right, so we've had some triumphs too. Smallpox officially has been eradicated, although it's still around in a few laboratories. Okay. Most of you have not been vaccinated. A few of you have been active duty service will have been vaccinated, but most not. And part of it was, as the number of cases went down by the early 70s, adverse reaction potential from this type of vaccine, a live vaccine, were greater than overall number of infections. And then another disease they've had pretty good success on is plague. All right, and of course with plague, we've had plague around forever. It wiped out, between about 1350, right? It wiped out 25 million Europeans, easy, a quarter of the population. Took 4 centuries for them to repopulate, all right? So again, but now we still have cases, anywhere from 10 to 25 a year, just not the same sort of outbreaks. And part of it is what? Antibiotics, which have been available since World War II. All right, this is what smallpox looks like. These permanent type lesions of the dermis. This guy, right, survives, he's going to have permanent scarring, but that's the least of his issues because this disease gets through all your organ systems. This is what a bubo looks like. All right. And again, what's a bubo? It's a lymph node that's been infected. It's full of pus, usually the bacteria there too. So in terminology, it's a swollen, in this case, inguinal, which just means your groin, lymph node, a bubo. We'll see bubos in several different diseases. Medically, it's a hemorrhagic suppurative lymphadenitis. So let's learn a couple terms here. Suppurative is a fancy word for pussy. That's it. Suppuration is the formation of pus. We'll take lymphadenitis and break it down. Start on the right. Itis just means inflammation. Word adeno means gland, and lymph, so it's a lymph gland that is inflamed. That's it. Lymphadenitis. All of you have lymph nodes you can palpate in your neck, your axilla, your inguinal region. They're all over the place. When they get swollen up, it's called lymphadenopathy, right? Just like the swollen. All right, so this is where we left off in the lecture when you get a chance to look at the first one. So we talked about here emerging versus re-emerging. So per your textbook, they're talking about re-emerging diseases as far as diseases that really haven't been prevalent in the last 35 years, but they're back. A re-emerging disease doesn't mean, or even an emerging disease, right, doesn't mean that it's brand new. It could have been around forever, we just didn't have the technology really to identify. So we used a couple of examples here Tuesday. First was tampons, okay? Now most of you are familiar with TSS, right? Toxic shock syndrome, right? And of course, this had to do with a very specific tampon, Relied. It was introduced in August of '78, and by January of 1980, there were about 1,200 dead young females, okay, nationwide. Has toxic shock been around? It's been around forever. But what was new was this polymer they used for this tampon. Because with this tampon, what you did is you inserted it and you left it for about 5 to 7 days. Okay? Craziness, right? But not at the time, all right? That was the hottest thing. So how do these people get this infection? Well, good old Staph aureus is in your nose, and about 5% of all Staph aureus carry this exotoxin, this protein, the toxic shock syndrome toxin, TSST. So exotoxins are what bugs make, their proteins. So as the woman either rubbed your nose in it early or it just fell out of your nose, I know it's sort of gross, but it happens all the time, right? Microscopically, it got in the tampon, you inserted the tampon, and there the bacteria grew like crazy on that tampon. The bacteria stayed put, and they secreted a toxin or protein into the blood, and the patient then, unfortunately these young, healthy women, average around 20, 21, got this terrible disease, toxic shock syndrome. Anyway, so the CDC tracked it and they realized it had to be this tampon, right? This is sort of end stage, worst case scenario, this patient died, and as the toxin gets into your blood, one of the things that can happen is your skin can desquamate or peel off too. Okay? But we'll get to this a little later in detail. Okay, so that was one example of like a disease that's been around, but it was re-emerging. Why? Well, because we had new technology, this Relied tampon. The other example we sort of closed class up with was Lyme disease. So, it's one of the bugs assigned this week, right? Borrelia burgdorferi. So, we talk about diseases, we'll talk about etiology, that means the cause. Okay, and I think one of your questions in the case study quizzes is identify the etiologic agent or the cause. So, how did Lyme disease come about? Well, it's been here forever too. What happened though is in the early to mid-70s, there was this cluster in Lyme, Connecticut, of these adolescents who were out in the woods doing what adolescents do, all right? And all of a sudden they had these big, crazy lesions that you know as a target lesion, okay? Or a bullseye lesion on different parts of their bodies. And about 75% of all patients present with that target lesion 2 to 3 weeks after what has to bite you for Lyme disease. A tick, right? It's a vector-borne disease. And it's a very specific type of tick. Genus is called Ixodes. But the tick bites you, And then as it feeds, and it takes, 24 plus hours easy, it doesn't happen in a minute, right? The tick feeds, it ends up injecting in a sense, right, as it's refeeding and regurgitating, it injects the spirochete, one of these bacteria called Borrelia burgdorferi. So, these kids had these infections, and a couple of doctors in that area, and Lyme, Connecticut at the time had a population of around 5,000, okay, it wasn't huge. But these doctors realized all these adolescents had these crazy target lesions, and they started digging around, and hence they figured out Lyme disease. It's been here forever, right? But they hadn't recognized it. Other things that come in for these emerging and re-emerging. Let's take a look at HIV. How long has HIV been around? 100 years at least, right? So what really happened? Well, it changed species, right? It went from chimpanzees, right? I went to medical school in St. Louis. They had a case of this young male from East St. Louis who was immunocompromised. He was a male sex worker, and he died from this horrible disease where his immune system failed. This was 1968. So they have his blood stashed. They saved it. And later, after early 80s, when they figured out what was going on, they checked his blood. What did he have? He had HIV, right? And there's other cases too. But really what happened big time was the virus jumped, right? Really from chimpanzees more into us with human, right? So we'll see evolution of infectious agents. Let's take something more relevant today. How about coronavirus? Where did COVID-19, SARS-2 come from? Well, we know it came out of Wuhan, China, but where did it come from there? Bats, exactly. The scientists went back to the same area of caves where SARS-1 came out of in 2000, 2001, right? So this coronavirus was normally found in animals. And that's why when you have a new virus that none of us have been exposed to before, because this usually is found in animals, it's called novel, N-O-V-E-L. And when you get a virus like that, or any sort of organism, you're going to get a pandemic, right? A worldwide infection. Because none of us have any immunity to it. So, sort of corona, this particular coronavirus sort of jumped ship from animals into us. All right. Okay, we'll get to corona in pretty good detail. Anyway, so HIV. Other things too, many of these bacteria, right, and the viruses too, a lot of them can share their DNA, right? And there's positive mutations and negative mutations. So let's take a look at some basic micro real quick. How long does it take, like a gram-negative rod, like Escherichia coli, E. coli, which everyone's heard of, to reproduce? To double. It's called doubling time, or generation time. How long do you think it takes? 10 months? That's us, right? Right? Man, we're on the tail end, people. We produce a new unit every 10 months. Now, how many years do you spend training them? 15, 20, right? Okay? Bacteria are what? And in 20 minutes in right conditions, they're dividing. So as they divide like crazy, there's a lot of, mutations that aren't advantageous, but the ones that are quickly get selected out. Okay? And these bacteria, right, also share, right, many of these mutations. Okay. Next. Other problem we're facing, resistant pathogens. Generationally, you hear about it in the news occasionally, right? About what? These, right? Superbugs that are resistant to everything that's out there, okay? With TB, malaria, gonorrhea, right? All sorts of stuff, okay. Right. Certainly we live in a global economy. It's great, all right? But, at the same time, there's drawbacks. It's older now. There was a movie, I think, called 12 Monkeys. Something like that, 12 or 13 monkeys, you ever heard of it? It's about a crazy scientist who gets pissed off the world, a group of people. What do? They brew up this really virulent, nasty virus. They hop in a plane and in 24 hours do a worldwide tour of all the major airports, okay? It's possible, this stuff now, right? You can be on a plane today, right, and 12 hours later you're halfway across the world. So, the example here is Haiti. They had a terrible earthquake in 2010. So, UN wanted to help, and they sent in Nepalese troops to help out. The problem was the water system was trapped. All the people from the rural areas moved into where? Port-au-Prince and some of the other cities. When they got there, the troops that were bivouacked out in the rural area, they have to defecate. The problem is the Nepalese have the highest carrier rate of cholera in the world, 15%, okay? So when the troops defecated into the streams, the water ended up going into the city, and you had in these quite tightly compacted groups of refugees, cholera spread like crazy. Because cholera is fecal oral, okay? When you get cholera, you lose about, worst case scenario, about 5 gallons of liquid stool a day, 20 liters. Dehydration kills you flat in about 3 days. Infants even quicker, right? But an adult can usually survive a couple days that way. So it doesn't have to be that bad, but worst case scenario, you can lose 18, 20 liters of liquid stool. Plus you could be vomiting, plus the heat. Anyway, so if you look back here, you know, Haiti is busy suing the UN right now, and Nepal, right, for damages, okay? But it's not really anybody's fault. Were they the best troops to send in? Probably not, okay? But they meant well, just what happened. All right. On this one, on 5, right, on changes in population here, okay? Right? When we talk about immunocompromised in this course, yes, right, we have an aging population, right? I want you all to be experts when you get out of here. When I'm worked out next door at the nursing home, I want you to know your stuff, right? Don't touch me otherwise, right? Okay? But aside from the geezers like me, what else you got to deal with? Well, yes, we have HIV disease still, and things have changed a lot in my lifetime when HIV first came on scene. And right, that progresses to ARC and then AIDS. But really, the third one there is the big one, right? Type 2 diabetes is a pandemic, and it's not really an infection, but face it, worldwide, type 2 diabetes is crazy, all right? Huge percentage of population. Why is that important? Well, as your glucose levels go up, the sugar coats your red blood cells, it coats the lining of your capillaries, it coats everything. And if you have poor sugar control, sooner or later your capillaries, they get sort of clogged off, and you lose blood flow. And as your blood flow gets down, your white cells can't get in, your tissue rots, and you get all sorts of infections. Okay, so diabetes is really a very immunocompromising type condition. Okay. So that's certainly a huge thing. Unvaccinated individuals, we had more of a problem with this from really starting in the mid-'80s up through, even recently. It's less so now. There was a lot of misinformation spread on vaccines for at least a decade and a half. Now, can you have an adverse reaction to a vaccine? Absolutely. Okay, but it's not what was pushed, right? And we'll get into the details on some of this later. Right? We have outbreaks in Georgia. We've had hepatitis A outbreaks. We've had some small measles outbreaks, right? I happen to be at UC California, right? University of California, San Diego, when the whooping cough kicked in 2010. We had lots of cases of whooping cough. Why? Because parents had not immunized their kids, okay? So, again, some populations, right, somebody can have an adverse reaction. But again, vaccines are to do what? They're to protect the mass, right? So the idea is you want to protect the route. You can get 90–95% of the population vaccinated. It's called herd immunity. And what that means, H-E-R-D, herd, is that if most of us are vaccinated, the 1% or the 5% that aren't won't get the disease because the disease can't get a foothold, right, in the population. And then lastly, just quick here, right, and some of the diseases we'll hit. You know, when I was, in medical school, I got my first ulcer, what the physician tell me? Well, this is like, late 70s, early 80s. He goes, no more booze, no more cigarettes, you can't swallow your chew anymore, no more fornicating either, okay, right? It's all out, right? Right? And he says, no. But what was the truth? What else was really caused by? Bacteria, right? Helicobacter pylori, okay? And that came out really in the late 70s, early 80s. Okay, toxic shock, okay. So, these PowerPoints are a mix of slides from Chapter 1, the intro, right? We're going to come back to a few of those in a minute. Chapter 16 on host-microbe interaction, HMI. And we're introducing that early because we're going to talk about terms like vector, fomite, right? Nosocomial, which just means hospital-acquired, iatrogenic, physician-induced, and a few other terms here. And again, really what I want to introduce you to in 16 is, normal flora, normal microbiota at different sites. So we've already covered a lot of this already. Again, our normal microbiota can change. Certainly, you pick up your first bugs at delivery, and that keep those for a while. Adolescence with puberty, there's some more changes too, right? And so some of our organisms are permanent. They're resident normal microbiota. You pick them up early on. Some of the first bugs you're going to pick up in your mouth are called Streptococcus, and you're going to pick them up from all those relatives who want to kiss your baby, okay? So, right? 6 weeks, people. Keep all those torturers away from your kids for 6 weeks, okay? Because everyone loves to kiss your baby, all right? But your immune system, how good is it? Not any good at all, right? So you pick up some bugs where you get delivered, whether it's home or at the hospital. You pick up bugs off people that hand you, but mainly your loved ones, right? Stuff like that. Okay. And then some of these are again, right? And so some of these you'll live with for life, right? And then others, right, are transient. Like, for instance, this pneumococcus. Next week, one of the assigned bugs is Streptococcus pneumoniae. Strepto means short chain. Coccus we're going to get to in a minute. It's just a morphology, a shape. We have cocci, which are spheres. We have bacillus or bacilli, which are rods. Okay. And we also have like spiral-shaped organisms as well. Okay. So Streptococcus pneumonia lives in our pharynx, our throat. What month is this? August still? I never know what month or year it is, all right? You just function, that's it, all right? Okay, all right? So come here in a couple of months, right? Respiratory season really kicks in. They don't even consider it in a sense, a primary pathogen in the sense that so many of us are really carry it, especially during the wintertime, and it's spread by respiratory problems, okay? So it's transient. Again, part of the winter area has two things factors involved. One, you're closer together usually, right? Huddled in, and two is the air is dry. It makes it easier to transmit respiratory virus, bacteria. HIV, all of you have been vaccinated for this. This is Haemophilus influenzae type B, okay? Again, another agent that used to be a huge cause, right, of epileptitis and some other diseases. And the meningococcus, if you live in the dorms, and I know the commons are open now, right, you have to be vaccinated for Neisseria meningitidis in order to go live there. Because it lives in your pharynx and spreads the respiratory. Okay. But you don't always carry Neisseria in your pharynx, but certainly at certain times of year people do. Okay, so these are transit, they come and go. All right, and then of course pathogens again are just microbes capable of causing disease. Okay, so we're going to blitz through a couple slides here. You don't have to memorize all the bugs in this list. We will repeat them, right? Ad nauseum in this course. You'll be able to sleep at night and dream of the normal flora, okay? You'll have them down pat. Now, we're going to focus on this course on medically important bacteria, right? So, for the micro majors, we'll have 3880 later that goes into a lot more on microscopes and a lot of other stuff. But we're going to pick up here much more on the clinical side of disease. We got to learn some basic micro, and then we got to throw in on top, get you up to gear with the case studies on clinical. So they're going to run through some different systems. Some of these bugs you'll recognize. So for instance here, and this isn't the greatest slide, right? If you take a look here on some resident microflora of skin and eyes. Well, there is Corynebacterium. That's a gram-positive rod. Corynea means club. When you look at them under a gram stand, they have a club shape to them. Right? Here is good old Staphylococcus. Right? Staphylum means bunch of grapes. Cocci means sphere. So when you stain these things, they look blue-purple, and they're a big clump together. Okay? Right, so you got lots of organisms here. Right? These are, again, you're going to find commonly on your skin. This is just another slide from Esther. Same thing. They're showing you different organisms. We'll get to all these over time. There's your nose, staph, Corynebacterium. Strep can live there too. There's your throat. All right. Never forget, right? We all have organisms in our distal urethra. Distal just means the farthest part away. So, I'll get a marker. But anyway, right? We got one. Thank you, Mr. White. I got one. Thank you. Okay. So when you worry about somebody having a urinary tract infection, you don't want to get the first part of your urine. They call it a clean catch. You want to be urinating and put the cup in because when they first urinate, they're going to take all those bacteria that are in the distal, the far end of your urethra, and they're part of your bugs that live there, and they're going to end up in a urine sample, right? You want to get a clean catch. Okay. Anyway, we got bugs all over us, people. Okay. On the skin here, just a real quick point. You can wash your hands with soap and water, right, which are great. You don't have to necessarily use a disinfecting soap, but part of with hand washing, the big thing, of course, is that you're going to take off any surface pathogen. You're going to take off, help desquamate dead skin cells. Then you can apply your ethanol-based Purell type stuff as well, right? But then in a short time, what's going to happen? The bacteria that live in your sebaceous glands and other places in your skin, they're going to repopulate. Things like Propionibacteria, Corynebacteria, staph, all right? They all come right back. So, we'll take a break in just a minute, right? Because we ended at 5, I think, right? So, for microbial pathogenicity, so we're going to be talking in this class a lot about pathogenesis, how these bugs cause disease. So, first thing all these bugs have to do is they have to attach to you, okay? So, what are they going to use to attach? Well, they have these structures called pili, also known as fimbriae. Many of them have a really thick, juicy capsule. Some of them have a highly irregular, loosely attached thing called a slime layer. Okay? So they have different ways, but until they attach, right at the very top, once they attach, then they colonize. And so how are you going to pick up staph if you don't have it? Well, you'll touch some fomite, inadvertently rub your nose, and Staph aureus, right, that Miss King's carrying, you're going to put it up your nostril. All right? Then what happens? Well, you don't touch it, but it's going to grow and colonize you. Do you have a disease yet? No. You're still okay, you just colonize. Now, right? However, what's escape host defenses? Well, maybe you nick your nose, shaving your nasal hairs. Who knows what, right? Okay? But once your skin, your integument gets nicked, that Staph aureus now can get through your immune system. When that happens, then they multiply more, and finally, you get damage to the tissue. Okay? And so it's disease. So you're colonized, it doesn't mean you have a disease until there's some other underlying skin trauma or something else. Disease, people, means your tissues are impaired, right? That's sort of the simple definition. You get damage. Okay. All right. So, okay. So, right, disease-wise, right? Again, we got this sort of a rule. This applies to bacteria, viruses, right? Protozoans, you name it. Okay? And so they're going to have to find a place to attach. So we're going to talk a lot in this course about a term called tropism. Right? So, they're going to have, these bugs have been around a lot longer than we have. And they have adapted, many of them, to attach to very specific markers on your surface. We have a glycocalyx too, a sugar shell, just like the bacteria do, just like the viruses do. All right? So let's take HIV. What people always hear about this, you've had some HIV in your other courses? Never? A little bit? Right? So people affiliate something called the CD4 count. You ever heard of that?
Yes.
Right? Well, CD4 is a marker certain of our cells have it. Right? The CD stands for cluster of differentiation. And they start from like CD1 all the way to CD gazillion, okay? They're just numbered, they're markers on our cells. They're glycoproteins that you and I have on our sugar shell. So in the case of HIV, what happens? They attach initially to a CD4 marker found on who? Macrophages, monocytes, okay, dendritic cells, and we'll get to all this in immuno. But they attach very specific, that's what tropism is about. Think of it sort of as a lock and key. They've had millions of years, if not billions, to evolve, and they hunt down a very specific receptor. Now, this CD4 receptor we utilize, okay? So they're pretty smart. They're not going to attach to some bogus receptor we can ditch. They're going to attach to something they need, that we need. And so, again, many of these bugs will be very specific. Another example I like to use is hepatitis. We're going to cover 5 types of hepatitis here by the end of the course. 2 of them, hepatitis A, hepatitis E, are fecal oral. If you have not been immunized for Hep A, could you get it leaving this classroom today? Absolutely. All you need is someone who pooped, didn't wash their hand, and left the microscopic fecal material in the fomite, the door handle, okay? You ingest that feces, you're not seeing it, right? You don't see it microscopically. About 5 weeks later, your liver gets inflamed. So what happens? You ingest that virus, replicates once in your intestinal tract, then it gets in your bloodstream, and its tropism is for what? Your liver cell, your hepatocyte. They cruise to your liver, attach, and then the disease starts, okay? So many of these bugs are going to have very specific markers. Part of our glycocalyx are going to attach to it. Okay, next slide. Okay, so we're going to talk in here about virulence factors, all right? So it's like a loadout, all right? Who here plays Call of Duty? I knew you did. All right, what are you playing most recently? Warzone. Okay, oh, she's really lethal, right? Only one survives with 100. I'm not playing with you, all right? You can find me online, easy.
Okay.
I play about 0300 at night, right? Geezer noob. You can't miss my gamer tag, all right? I suck, by the way. You want me on the other team, right? I have a kill-death ratio of like 0.001, all right? Okay. Anyway, all right, so what do you have a loadout, right? Don't you have your favorite loadout?
Yes.
All right, so maybe she's packing, what do you carry for an assault rifle? Or you're a sniper? No. All right. You're going to have what? She's going to have her favorite rifle. She's going to have some body armor. She's going to have some attachments like a mega scope, right? All sorts of stuff. That's what virulence factors are, all right? The more virulence factors you have, the baddest ass you are, okay? You're a bad mother, right? And that's how the bacteria are. Some of them are loaded to bear with virulence factors to kick your butt, okay? Like staph. Staph warriors potentially can carry lots of them. Others may only have one, all right? So let's take Staphylococcus epidermidis. That's a bug that lives on your skin. So this bug, right, very prominent, about 55% of the bugs on your skin, and there's lots of bugs that live on your skin. Its main virulence factor is called, basically, a slime layer, okay? It doesn't have lots of nasty toxins. What happens in real life? Well, you get a catheter put in you, like an intravenous line, and it's not by purpose or choice. Your last name is?
Kelly. Kelly?
Last name's Kelly? So Miss Kelly over here decides, you know, she's putting a line in you, but she's sort of looking at her hand, maybe didn't clean your skin quite as good as she needed to, right? Didn't disinfect it. The line goes in, that catheter picks up a little staph epi off your skin. What's going to happen to that line? Well, that catheter, people, is a fomite, an inanimate object. Staph epi, which is not normally a nasty bug, gets on that catheter. That slime layer lets it attach to that piece of plastic. It then forms a biofilm, all right? We'll get to those. It secretes one. And about 3 days later, if not sooner, you have these really red lines running up your arm, right? And Dr. Kelly has to come in and pull your line and put it in the other arm. Was it on purpose? No. Well, should you sue her? Yes. Okay? Right. It just happens. But again, Staph epi has one big virulence factor. That's slime layer. It means a fomite too. Other bugs, again, like Staph aureus we talk about, will have pages, okay? They can nuke you from any which way. So virulence factors are key in this course. So all of them, of course, got to attach. They got to have some way to attach to you. Right? Once they attach, again, they got to colonize you. They're going to reproduce, and then some of them make toxins. So early on, we'll come across this term called exotoxins, and these are proteins, like we saw in toxic shock syndrome, that some of these bacteria secrete. Some other bacteria, right, called gram-negatives, have another compound they make, right, which is called endotoxin. So, endotoxins are also known as LPS, lipopolysaccharide, right? And that's what they are. They're part fat, part carb, right, basically. Exotoxins, people, are proteins, okay? Now, do all bacteria make exotoxins? No. Some gram-positive do, some gram-negative do. As a general rule, all gram-negatives have LPS as part of their cell wall. Okay. So again, some of these virulence factors are chromosomal. And again, bacteria as a general rule have one supercoiled, double-stranded piece of DNA. We'll get to exceptions. And then they have extrachromosomal. And again, these are what? Small, circular, double-stranded pieces of DNA called plasmids. And these plasmids usually, right, confer on the bacterium some advantage. Antibiotic resistance, disinfectant resistance, right? Some beneficial fact. And these bacteria, of course, can share these plasmids, right? Virulence factors with other bacteria. Okay, so we'll stop here, right? This is just showing you a bug attaching. There's its specific receptor it wants to, okay? There's the adhesin again, right? So these bacteria, whether it's a pili or a fimbriae or whatever it's using, some of them have little proteins on the tips called adhesins. They'll help attach to a very specific receptor on your cell, all right? And we'll go from there. Okay, so we'll come back here, we'll run through these definitions, and we're going to move into gram state today, and we'll have plenty of time to do that. Before I hit pause on this, any questions for me? Okay, I'll see you back in about 5 minutes. Our regimen isn't perfect here with once every couple of weeks. In fact, I won't see you probably for a little over that. But you're always welcome. I'm on campus Tuesday, Thursday, easy, 12, 13 hours plus. I come in at 0700. I'll be here tonight till 19, 2000 at 7, 8 p.m., right? So make an appointment. I have office hours right after this class. If you have questions, ask, okay? You're not alone. Okay. For emails, I really prefer you email me on iCollege. And the reason being, my other GSU email, mburnett@gsu, gets deluged with what? University stuff. It's easy to get buried. On iCollege, it's just students, so and I check that all the time. Okay, everybody knows what a fomite is. Inanimate object, right? Their desks, right? Computer keyboards, Generationally, what's one of the filthiest things you carry around with you all day? Cell phone. You don't see me carting one, no way, right? Okay, right? Cell phones, right? Okay. All these things are fomites. Of course, the vector is what? What's a vector? Fomites are inanimate, vectors are animate, right? Mosquitoes, insects, right? Arthropods, right? And we'll see with the vectors, we have mechanical vectors in chapter 16 and biological vectors. So mechanical vector means, I'm going to break my neck on this semester. There. I'm one of those kids that was fidgety all the time. I can't stay still, right? All right. All right, so with mechanical vector, what happens? Right? So we have a fly, right? And it was Fareed. She leaves here and goes out down the street and eats a burger outside. Okay? And, and the fly, Freddie the fly, lands on her burger. But before he landed on her burger, where was Freddie? I mean, he was hanging out down the street on some wonderful feces, okay? He picks up this nasty bug called salmonella, lands on Miss Freed's burger, she chomps down, right? She gets salmonella, all right? So, that's a mechanical vector. The bacteria was picked up on the outside of the fly, landed on her food. Then there's a biological vector. This would be like malaria. The mosquito, right? The mosquito has a couple roles here. But in that case, a biological vector means that the organism undergoes, right, development, maturation inside that host. Mechanical versus biological. Mechanical vectors basically transport the bug from A to B. Biological vectors, like we'll see, is with Lyme disease, right? Because remember, in Lyme disease, inside Ixodes, Borrelia, the tick, Borrelia is reproducing. Okay? All right. Next, chemotherapy. Generationally, when you hear the word chemo, what you think of is cancer. Okay? Now, have any of you taken antibiotics in here before? Yeah, you've had chemotherapy, right? Just general term chemo means chemicals. So, yes, there's chemotherapy, again, oncology, right, for cancer. But there's lots of different other chemotherapy. Antivirals, antibiotics, right, antiprotozoal drugs, just in general. Okay. There was a question on your first quiz last night that asked you to choose all of the following terms related to morphology except one. And one of them was a genus you've probably never seen before, but it was italicized, first letter capitalized, okay? Right? So we're going to deal with here genus and species, that's it. Not the rest of that stuff. It'll all be memorized some, what, mnemonic from biology, early biology, what is it? Kingdom, phylum, I don't know. You think I know? No way, right? Okay? I deal with genus and species, man, that's it, all right? But that's all you need to know in here. But you got to know genus is capitalized, right? And italicized, or capitalized and underlined. Okay, all right. So, virulence factors, right? You're an expert on now, okay? So in this course, we're going to talk a lot about pathology, virulence factors, how these bugs cause disease. Again, pathogenicity just relates to, again, right, how nasty that organism is. And that partly ties in with how many virulence factors you have. All right? So for instance, one of the bugs here, right, all right, we got two bugs here. First is Streptococcus pneumonia. Okay, so we're going to talk a lot about a lot of different types of Streptococcus here. Strepto means short chain. Coccus, of course, is a sphere. Strep pneumo, they have about 90 different capsules. Right? Remember, capsules were firmly attached, right? Sort of, right? Very organized structures made of polysaccharide, carbohydrates. Since there's around 90 different capsules, how many times can you get strep pneumonia? 90 times, right? Most of you have had 2 vaccines. One of them covered 13 strains, another may have covered 7, another one may have covered more, but there's still 70 more waiting to get you, okay? Each time you get one of these Streptococcus pneumonia, you're going to make antibodies to it. The point is with strep pathogenicity, if it doesn't have a capsule, it can infect you. Strep pneumo, no capsule, no disease. It's a key virulence factor. And they use that capsule as you spread it to respiratory droplets to attach right to your throat and then later into your lung. Okay. Another bug with pathogenicity here is good old Neisseria gonorrhea, right? To cause, right, etiology to cause it, right? Gonorrhea, clack, okay? And so for Neisseria, they're coated in these things called pili, right, or fimbriae. And they're very similar. They use those pili to attach to your mucosal membranes, your pharynx, your eye, your urethra, your cervix, right, your rectum. If they don't have any pili, they can't attach. No pili, no disease, right? It's a key virulence factor for them, part of their pathogenicity. Okay, term ones, virus, viroid, and prions. That's sort of the tail end of chapter 1. So, viruses are obligate intracellular parasites, okay? So when they're out cruising around, right, in the air between us, all right, and so, right, this is Mr. Guy, right? Okay, so there's viruses hanging out near Mr. Guy in the air. They're inert, right? I-N-E-R-T, right? They're like Master Chief in Cryo, okay? He's hanging out taking a snooze. But the second you inhale them and they attach to your cells, things change, all right? They attach to your cells, we bring them inside, all of a sudden they're alive. Okay, so viruses now, they come in a couple different types. They have either an RNA genetic base or a DNA, but not both. Okay, we have both RNA, DNA, so do the bacteria. And the viruses also then have a capsid. It's called a protein coat, capsid, made up of capsimers. Viral protein coats self-assemble, right? They don't need a ribosome to put them together. And then some of the viruses, okay, have an envelope on the outside, all right? They get that envelope, not all viruses are enveloped, some are. Enveloped viruses, right, they get it when they exit your cell. They pick up your cell membrane. There's a couple of exceptions, but as a rule. So viruses are either RNA viruses, DNA viruses. They can be single-stranded, they can be double-stranded, they can be naked, means a protein coat, or they can be enveloped, which means a protein core with an envelope on the outside. Okay? So, everybody loves herpes virus. It's everybody's favorite. Okay? Herpes is a enveloped, double-stranded DNA virus. All right? So in the emergency room, they have a great saying, right? What's the difference between love and herpes? Herpes lasts forever.
Oh, wow.
Okay? Right? Absolutely. All right? And the herpes virus that you pick up, where did most of us get our first herpes viruses from? By the time you were 3, you picked up HSV1, almost all of you did. Where'd you get it? Your loved ones, right? They're slurping all over you, all right? You pick up lots of herpes viruses that way, right? Now, if a kid picks up HSV2, something else is going on, right? Okay, it's a general rule, all right? But many of us picked up cytomegalovirus, all these viruses we'll learn about that are herpes viruses, okay? But they're an enveloped virus, a class of them. We'll cover a bunch of them. What about viroids? Viroids are just a single-stranded circular piece of RNA. Generally, we won't cover them a lot. We'll talk about them a little bit today, and that's about it. They really infect plants. The closest disease that humans have related to a viroid is hepatitis D, HDV. It's not technically a viroid, but it's sort of similar. In your lifetime, probably things will change, but right now we just find them in plant disease. But again, it's a single-stranded RNA. They're obligate intracellular parasites. Just like those viruses hanging outside Mr. Guy there, outside is, hanging in a space around them, they're inert, doing nothing. Once they get inside you, though, and get inside a cell, now they move on, they're alive. Okay. Lastly are prions. And these are proteinaceous, infectious particles. So, prions are abnormally shaped proteins. Now, every one of us in here, in our nervous system, in our neurons, have prion proteins. We have the normal prion proteins. What happens is, there's different ways you can get these diseases. About 1 in 7 million or so people genetically are predisposed to develop, at some point in their life, these prions change shape, and they become abnormally shaped. So remember, prions have no RNA, no DNA, they're just a protein. And so when the protein changes shape, and what it does is, over time, slowly, it gets neighboring prion proteins that you have that are normal to change their configuration, change their shape, and they become abnormally shaped. As these proteins become abnormally shaped, they form, right, sort of fibrils, and those fibrils precipitate out a clump. They're called amyloid plaques. You've ever heard of Alzheimer's disease, which is a dementia? People get plaques with Alzheimer's. These aren't quite the same thing, but it's similar. You get these proteins, they deposit out inside your brain cell, your neuron, and what happens? The cell dies. When the cell dies, you'll see these giant holes. They're called spongiform, right? Look under a scope, they look like a little bit of a sponge. And the other term is spongiform encephalopathy. Encephalo, again, is brain, pathy is disease. All prion diseases are fatal. Now, you will know of a prion disease, too. You know it as mad cow disease, right? Came on the scene big time in 2000. right? We were much younger then, right? But Britain wiped out their entire cattle supply over that. Because what happened in that case? It was the first time a prion disease from sheep ended up in some of the cattle feed, and from there it got into humans too. Okay? The Brits like this brain soup. It's a pretty common meal over there. And so they had a bunch of young people your age getting, right, prion disease, not normal. If you're genetically predisposed, it's called Creutzfeldt-Jakob disease. CJD usually kicks in by 45. Again, all prion diseases are uniformly fatal. There is no treatment, no cure. Okay? And then the last way you can get prion diseases, how they were historically first discovered, right, was good old kuru. How do you get kuru? You guys know? Ever heard of it? I'm going to take Mr. Guy hunting with me at my home state, Alaska, right? We'll go up, right, in the winter. I don't want this to happen to him, but, you know, if things go south and he doesn't make it and we get stranded, I'm going to eat him, okay? No sweat. He's a nice, big, hulking guy. He's going to keep me fed for a couple days, all right? It's nothing personal, all right? Of course, Mr. Guy, what's he got waiting for me? He didn't tell me that he happened to have eaten mad cow, right? Okay? So I eat his brains, what do I get? I don't have to use his brains, right? So kuru was discovered in cannibals, all right? New Guinea, all right? And what would happen is, one of the things they do is when the person died, they'd serve them up, right? Especially their brain to all the relatives, okay? So initially, that's how they discovered the prion diseases. Don't worry, I won't eat you right away when you're alive. Okay, wait till you're gone. All right, next. All right, so that's prions. Okay, let's move on to Gram-stain decay. This is the most important slide out of chapter 1. They're going to take you and break you down into domains. So we have the living organisms and the infectious agents. Okay, so you have the prokaryotes. Remember, pro means pre, karyo means nucleus. So they have, right? They don't have a true nucleus. That's the bacteria and the archaea. Archaea means ancient. After today, we'll never see them again. That's not what this course is about, all right? They have pseudopeptidoglycan. You'll see that soon enough, all right? They don't really infect us because they live in extreme environments. Again, probably in your lifetime, we may come across some archaeal diseases. And then we have the eukaryote. EU means good, normal, true. Karyo means nucleus, okay? So the eukaryotes have a true nucleus. So you break, these are the domains, bacteria and archaea lump into prokaryotes. Now, these groupings aren't perfect because we have more in common with the archaea than the archaea have in common with the bacteria. But that's for an evolution course, okay, later. So this is prokaryotes, over here eukaryotes. Of course, the euks break into algae, protozoa, right? If you like protozoa, I teach a parasitology class, 6,000 level. We spend half the course on protozoa, it's parasites. Fungi, helminths, of course, are multicellular parasites. Everybody, tapeworms, roundworms, all sorts of great stuff, flukes. Where are humans at in this chart? Where do we belong? Right? It's easy to forget. You need to make another box right over here. Put a box and put me, okay? Because we're eukaryotes, all right? Don't forget. Okay. Protist, forget it, right? Obsolete term. If anyone uses it, man, avoid it, all right? Okay. It was a catch-all category. If one of the bugs didn't fit in some of the other categories, well, they're a protist, all right? Really not used so much anymore. Okay. Over here, we have the infectious agents. So, by definition, okay, and I always say this, we can spend the semester in a philosophy course discussing if viruses are alive or dead. But by definition, if you're acellular, A means without, AN, right, acellular, one word, then you're non-living. That's viruses, viroids, and prions. Viruses and viroids are obligate intracellular parasites that got to get inside your cell. Prions are not. And part of it is, with prions again, yes, they live inside your neural tissue, but you're going to eat them sometimes, one way to get them, right? They're going to pass through your system, so they're not, right? Viruses, man, they got to reproduce inside your cells. Okay. All right. So, you have videos. Now, most of you have Connect. Please, I posted for the homework, right? There's some videos attached too. But take a look. They have tons of great videos on Connect there. You can take a look after each of the chapters. If you look over at supplemental material, this is why I made this great list, right, of all these videos. Unfortunately, they copyright struck the website, okay? But you still have the same videos. She has all wonderfully listed out for you that are available on Connect. They're all McGraw-Hill, okay? All they did was, about a month ago, they copyright struck this one website so that you could see them for free. Now you got to buy them on Connect. But they really are very good. So take a look at some of these. We'll look at some next class too. Okay, So I want to spend a couple minutes here on prokaryotic cell before we run out of time, and then gram stain. So what you're looking at here, right, is sort of a classic, what shape is this cell? Bacillus, right? A rod, exactly, okay? And so structurally, on the outside here, we have their capsule. Again, capsules are firmly attached, sort of a regimented structure. They're almost always, right, they almost all are polysaccharide. There's one exception. That's a bug called Bacillus anthracis, okay? All right? The rest are all carbohydrate polysaccharide. Inside here is the cell wall, right? There's two sort of solutions to the cell wall. One is called gram-negative, which is also known as a diderm, and the other is a monoderm called the gram-positive. Inside that's the cell membrane, and then inside here is their nucleoid. That's their single double-stranded supercoiled piece of DNA. They don't have a nucleus, okay? So it's a nucleoid, and they got some ribosomes, maybe some storage granules. If they're motile, they have a flagellum. Never miss on a quiz or test if they give you the option of cilia. Bacteria don't have cilia. Only eukaryotes have cilia. Bacteria don't, right? Now, if a bacteria is non-motile, it means obviously they don't have a flagellum, okay? Right? That's how they're going to get around. Okay. All right, so quick overview. So this is showing you a gram stain. Right? Remember, these stains are great. Why? Well, bacteria are normally colorless. If you didn't have stains, all right, and we're going to use a couple stains in here, crystal violet, saffronin, methylene blue, a couple of these, malachite green. These stains increase the contrast, so you can see them. So one of the questions on your first day quiz, right, was about what can you see when you stain these things? Well, when you take one stain, let's take crystal violet, because that's the first part of the gram stain. You take your smear, you take a specimen, right, you put it on a glass slide, you're going to put a Bunsen burner to it, you're going to fix it. You put crystal violet on top for about a minute, all right, and you wash it off. The crystal violet is called a primary stain. And what will show you is the morphology. It will show you whether they're rods or cocci or whatever, but it's not going to help you differentiate the two different groups. So after you put the crystal violet on, what do you do next? Well, then you add this stuff called Graham's iodine, also known as the mordant, M-O-R-D-A-N-T. And that mordant is going to combine with the crystal violet. Okay, and when it does, it forms this great complex, and it's going to get trapped underneath this bacterial cell wall, which they show here in blue. Gram-positives have a really thick cell wall, 30, 40 layers thick. Gram-negatives have a thin cell wall, 1 to 3 layers thick. So at the end of step 2 of the gram stain, you're going to have your crystal violet and your iodine trapped. The next step, the third step, is called the differential step. And it's called that because when you add the alcohol acetone mix to that slide, it's going to separate the bacteria into two categories. So the acetone alcohol mix against the gram-positive, what it will do is cause this thick layer of protein and sugars, peptidoglycan, to basically become dehydrated. The crystal violet iodine gets trapped inside, and the bugs retain that blue-purple color. The gram-negatives, because they have a different cell wall setup, they're going to have a thin layer of peptidoglycan. And then outside of that, they have what's called an outer membrane. Acetone alcohol mix trashes the outer membrane, and as a result, the crystal violet iodine seeps right out of the cell. So at the end of the third step, the differential step, the gram-positives are blue-purple, the gram-negatives are colorless, okay? The fourth step is the counterstain, and that's saffronin. Saffronin is sort of pink-red. You put saffronin now on for about a minute, 45 seconds right there, all right? What happens? Well, the gram-positives suck up the saffronin too. But the crystal violet is so intense, the blue-purple overshadows whatever red dye they pick up. The gram-negatives were colorless. You've now added saffronin, all the gram-negatives stain, pink-red. Okay? The gram stain is a differential stain. Helps you split them into two groups, GP, right, and GN, right, basically. The key step is the third point. When you add the alcohol acetone, it's the differential step it's going to separate. Okay, so your classic gram-negative rod here. Right, so again, most of them are, many of them are covered in pili or fimbriae. In this case, it's got one flagellum. There's their nucleoid. So this is a couple of clinical infections coming up here, bugs will cover. This is called a carbone, right? Okay? So, what is this? You know what the initial lesion, what this is, it's called a boil. That's also called a furuncle. When you get a bunch of furuncles together, like this person has, all interconnected underneath, you get a carbone. This is Staph aureus, okay? So what do? You have to wait till this thing gets nice and juicy finally, and then you take a scalpel, it's called I&D, you incise and drain it, okay? All right, we'll get to all that. All right, this is what most of your toenails look like, I'm sure, right? Okay, not yet. Wait 50 years, all right? At that point, your immune system is finished, you got fungus all over you, right? This is a onychomycosis, classic sort of fungal nail infection. All right. This is a protozoan bug. This is called Trypanosoma cruzi, known as Chagas disease. Right, so these live in our bloodstream and other places. And back to, again, chapter 1, viruses, viroid prions, we already covered these. Nestor has some great tables. Use them. Help summarize key facts like here, right? OIP, right, exactly. So take a look at them. This is just showing you how viruses are made. There, in this case, this is an RNA virus, similar to COVID. COVID-19 is an RNA virus. And we'll see it's made up, it has a protein coat made up of capsamers, which all together form a capsid. And of course, with SARS-2, the difference is it has on top of it an envelope. All right. And on that envelope, they're going to have these glycoprotein spikes that are all at one end, which gives it the crown, the corona, and that's what they use to attach to your mucosal epithelium or into your lung tissue. We already covered viroids, again, single-stranded RNA. Okay. There's prions, right? There's Nestor's Micro has a great video on prions. Take a look at it. We'll walk you right through. This is figure 1421. We'll walk you through to the conversion. Right? So you definitely take a look at it. We'll come back to these in the neuro section. Right? And as we wrap today here in the next couple of minutes, so here is our demonstration of a, right? Take a specimen, they say air dry, right? We're going to fix it with heat. We add our primary stain, in this case crystal violet. It's great for giving you the morphology, right? Cocci, rod, whatever, but it doesn't tell you squat about whether they're gram-negative or gram-positive. Okay. So again, we went through this. All right. And this again is the gram stain I just talked you through. Everybody stains purple. We add the mordant. Everybody stains purple again. Then we add the differential step. And again, if you're gram-positive, the crystal violet iodine gets trapped. If you're gram-negative, the alcohol trashes their outer membrane and the stuff escapes. At the end of step 3, the gram-negatives are colorless. When you add the counter stain, key point, it stains both gram-positive and gram-negative, okay? Just the gram-negatives show up pink because they were colorless. All right, so you look at this smear initially and you think, what a mess, right? This is like one of those Rorschach diagrams they show you at the psychiatrist's office, right? Tell me what you see, okay? But you'll get better. You'll figure out how to look at these things. All right. Take a look at some of the morphology. Again, so coccus is sphere, bacillus is a rod. They can be in between. Coccobacillus, that's just great, all right? Some of them are curved. Vibrio means comma, all right? So the genus that causes cholera is Vibrio, and the species is cholerae. It's shaped like a curve. Some of them have a sprig like a spiral shape, spirillum, and then of course some of them are spirochetes, sort of like a slinky, tightly coiled. That's diseases like Borrelia, or like syphilis is another one, similar like that. Okay. We'll stop here, right? I'll hang for questions, right? Anything, any questions? Yes?
You mentioned that these viruses attach either DNA or RNA, but since RNA has 4 different types, does it matter which type it attacks?
Well, so what they mean is their genetic material is going to either be RNA or DNA, right? So for us, ours is what? Our main genetic material is double-stranded DNA. We have 23 pairs of chromosomes, all right? So as we get more into virology, you'll see some of these viruses will have a very small piece of RNA. Some of them will have several segments of it, but that's their genetic code, right? So it's not like you're thinking like transfer RNAs and some of these other nucleotides, but their whole genome is RNA-based. Just it, or DNA, right? But never both. We have both, right? We have DNA as our base, and then we do what? We make messenger RNA, right? And from that we make proteins, okay? And so what you'll see when we get to some of these RNA viruses, like coronavirus, is when they get into your cell, they're going to use your machinery, because in minutes, when a virus infects your cell, they own you. They take over your machinery. They're going to use your setup to make messenger RNA, make proteins, and make more virions. But we'll see the RNA viruses bring with them special enzymes to reproduce their RNA gene. Yes?
The mordant on the board refers
refers to the second instead of the first.
The mordant is the iodine, called grams iodine. That's the mordant. And what it's going to do is attach to the crystal violet, and that complex then gets trapped in gram positives. But in gram negatives, it's trapped initially too until you add the third step, the alcohol acetone, and then it gets released.
So iodine and mordant are the same thing?
They are. The mordant equals grams iodine.
Oh, okay.
Right? So when they say mordant, that's the grams iodine in the gram step. The gram step. You're welcome. Other questions? All right. So